Protein perform many functions that are essential to life. Proteins act as an enzyme, hemoglobin that transports oxygen, antibodies that fight infections, and many more. Chromosome is a thread-like structure in the nucleus where DNA molecule is found. Chromosomes carry genes. These genes code to a specific protein. So how does a gene get out of the cell nucleus to make protein and do its specific job? The first step in the protein synthesis is called transcription, which occurs in the nucleus. DNA serves as a template for mRNA formation. The strands of DNA are antiparallel. One strand is called the sense strand, which is found in 3' to 5' direction. This is the coding strand that gets read. The other strand is called the antisense strand. Antisense strand serves as a template for mRNA formation. RNA polymerase attaches to a promoter and unzips the DNA helix so that complementary base pairing can occur. Complementary base pairing takes place similar as DNA replication. Cytosine always pair with guanine. The main difference in the DNA, adenine always pair with thymine. However, in RNA, there is no thymine. Instead, there is uracil which replaces thymine. Once it unzips, RNA polymerase brings the free-floating mRNA nucleotides to complementary base pair. Template strand. RNA polymerase builds RNA molecule in the 5' to 3' direction. A single strand of RNA called mRNA transcript is now formed, which contains the gene information. Now, mRNA must be processed before it leaves the nucleus. One is called the introns, which are junk genes that don't code for a protein, so it needs to be removed. The other gene is called exons, which get to exit the nucleus. Thus, a mature RNA transcript is formed. mRNA has a 3' end called a poly A tail nucleotides added to the end. The other end is called a cap, located at 5' end, which is composed of an altered guanine nucleotide. Now, a mature mRNA transcript can leave the nuclear pore and goes to the cytoplasm where it is attached to a ribosome. Now, mRNA is attached to the ribosome, which is composed of ribosomal RNA found in the cytoplasm where translation occurs. During translation, there are 3 steps. First step is called initiation, in which the small ribosomal subunit binds to the cap of mRNA and moves into the tRNA initiation site. mRNA codon pairs with the anticodon of tRNA. The start AUG pairs with the anticodon UAC. A large ribosomal subunit joins the small subunit to create the 3 binding sites for tRNA. Amino acids or anticodons of tRNA are matched to the codons of mRNA in the A site. The ribosome moves along the mRNA. tRNA exits from the E site and the next tRNA enters the ribosome. This process is called elongation, as the tRNA is positioned at the A site. The peptide chain will be transferred to this tRNA. This makes the amino acids longer by adding tRNA from the growing chain in the A site. This continuous process leads to the peptide bond formation. When a stop codon is encountered at the A site, termination occurs and a ribosomal subunit dissociates. A chain of amino acids, or what we call polypeptide, is released. The endoplasmic reticulum carries the newly synthesized proteins to the Golgi body. Golgi body will modify the protein and enclose them into vesicles. Now, a specific protein is produced and is ready to undergo structural organization. The final shape of protein determines its function in living organisms.